and welcome to this lesson for higher biology, which today is looking at enzymes and metabolism. So up until now we have been looking at how metabolism actually acts in a cell, what it does and where it's occurring. What we're going to be looking at today is the role of enzymes in controlling that metabolism. And so what you're expected to be able to do is explain enzyme function in terms of the induced fit model. You need to be able to state the relative affinities of an enzyme for its product and its substrate. You need to be able to describe the activity of enzymes in terms of its substrate and enzyme concentrations and how those can affect the activity of an enzyme. So first of all, quick recap from last time that enzymes are going to be found in the cell in the cytoplasm, on all of the membranes, and in and around the membrane organelles such as lysosomes, mitochondria, nucleus, chloroplasts, etc. And that those membranes are helping maintain the correct enzymes in the right locations in order that these reactions can happen as quickly as they need to be able to. The purpose of an enzyme is for breaking down or creating bonds in reactants, and initially we have, if say for example we have a reaction where we're creating a product from two small substrates, we've got our substrates binding with our enzyme, forming our enzyme substrate complex, and these are forming our product molecule. So initially they are in our reactant state before they eventually turn into our products. And one of the things that enzymes are doing is helping breaking those initial bonds in the reactant. So they're changing the bonds of whatever chemicals are involved in each molecule to provide us with molecules that are in a transition state. And when they're in this transition state, the enzyme is then able to form new bonds and produce our product. So enzymes are essentially serving to lower the activation energy which is needed for a reaction to proceed. So if this is the amount of active energy that we need, and this is the amount of time that we've got available to us, the enzyme acts as like getting this boulder over this hump. So if the reaction is going to take place, once it's gotten to a point, it's able to take place. We have kinetic energy of motion in this one. So, and similarly, we have energy available so that the reaction can take place. What the enzymes are doing is lowering the activation energy, lowering the point at which this is going to occur, and allowing us to get the reactants up and over the hump in order for that to happen. Now, when an enzyme meets its substrate or substrates, it may not be an actual physical match. Its shape might be slightly different to the active site of the enzyme. However, as the enzyme and substrate start to bind, we have something called the induced fit model, and that's where our active sites are molds around the substrate, making sure we've got a really tight, snug fit and that we've got as much surface area contact with our enzyme and substrate as possible, until eventually our substrate is changed shape, and that allows the enzyme to release the products. Now, in terms of affinity, enzymes have what we call a high affinity for their substrate. That really just means that they've got a high chemical attraction to their substrate molecules. So they're more likely to bind onto their substrate than they are to bind onto any other molecule. That means should the enzymes and the substrate be in a similar location, they are more likely than not to bind with each other. However, enzymes have a low affinity for their product, which means once the product has been formed and that substrate molecules have changed shape to form our product, it's immediately released into the environment, leaving our active site free for another reaction. So enzymes have a lower affinity for the product, meaning that they don't want to bind to that, they're not interested in it. Once our substrate has been formed into the product, it's going to be released into the environment. That allows the product to get to wherever it needs to be or go and join with the rest of the pathway that it's occurring, and it also allows the active site of the enzyme to be available for another reaction to happen. Now, there are a few different factors that are able to affect enzyme action, most of which you've already covered at National 5. And there's a few others that we're going to mention just now. So we need to discuss the fact relating to temperature, pH, and the impact of substrates and enzyme concentrations themselves. Enzyme action can also be affected by whether or not there is an inhibitor present, and we will come to these later on in the course. So to begin with temperature, as you'll have seen at National 5, as the temperature of our reaction increases, so does the activity of our enzyme. That's as a result of the amount of energy available in the reaction. As the molecules are moving around a lot faster, they're more likely to meet and for that substrate and enzyme to bind and form that enzyme substrate complex to start reacting. When our enzyme meets its maximum rate, that's our optimum temperature. After the optimum temperature, however, the structure of our enzyme is damaged or denatured and the activity is going to drop off sharply. Now you see this in lots of different organisms in terms of the sort of shape of our graph. So we've got a very similar curve for psychrophiles, mesophiles, thermophiles and hyperthermophiles. However, their optimum temperature for their enzymes to operate at is quite different. So it's worth recognising that although you'll see a similar curve, it very much depends on the type of organism as to what sort of temperature might be our optimum. The ones that we're used to talking about are mesophiles, where we're talking about 37, 38 degrees-ish normal body temperature. However, different sorts of organisms are going to have different appropriate optimum temperatures. Now, in terms of our substrate concentration, if we have too low a substrate concentration, there are too few substrate molecules present to make use of all of the active sites on the enzyme molecules. So there's not enough substrate around. When we increase our substrate concentration, however, we start increasing our rate of reaction until we get to a certain specific point. And that point is going to vary depending on the concentration of enzymes. Once we've increased our substrate concentration, we're increasing the use of those active sites and how many are available. If we get to a certain point, though, where we're increasing our substrate concentration and our rate of reaction is not continuing to get any faster, that means that all the active sites must be occupied by the substrates already present, so that the enzyme concentration itself has become a limiting factor in that instance. Where we have enzyme concentration reactions, when we look at those, we would need an unlimited supply of a substrate in order to have a graph that would look like that in terms of our enzyme reactions. It's very unlikely that we would have an unlimited supply of the substrate, as they're usually being broken down quite quickly, and we don't always have an unlimited supply of anything in a cell. So the enzyme concentration graph would be directly proportional to the rate of reaction. The more enzyme you've got, the faster the rate of reaction is going to occur. However, this would only be the case if we had that unlimited supply of substrates, which is very unlikely to happen. So you should be able to explain that enzymes function in terms of the induced fit because the active site is able to mold around its substrate, producing that induced fit model and allowing an increased level of surface area contact between the enzyme and the substrate. That the enzyme has a higher affinity for its substrate, meaning it's more likely to bind to that than anything else, and quite a low affinity for its product, allowing the product to be released into the environment and leaving the active site free for further reactions to take place. That the activity of enzymes in terms of substrate and enzyme concentrations are going to be quite different depending on the other factors within that reaction. That as you increase your substrate concentration, you will also increase your rate of reaction until such a point as enzyme concentration becomes a limiting factor. So increasing your substrate increases rate of reaction until we run out of enzymes available. As you increase enzyme concentration, however, you're able to see an increased rate of reaction, and that will continue as long as you have an unlimited supply of a substrate, which is quite unlikely. So, as ever, feel free to ask any questions on this as we go forward in class.